The overall goal of the following experiment is to show non-adherent effector T lymphocyte migration over a monolayer of adherent tumor cells. This is achieved by first establishing a monolayer of adherent tumor cells in the wells of poly-D-lysine-coated Teflon masked slides. As a second step, fluorescently labeled effector T lymphocytes are sedimented into the center of the monolayer using a cell sedimentation manifold. Next, light and fluorescence microscopy are used to visualize the migration of the non-adherent cells over the monolayer. The results show that the migration and effector functions of non-adherent T lymphocytes in co-culture with an adherent tumor cell monolayer are maintained after the lymphocytes have been transduced with a retroviral vector.
The main advantage of this technique over existing methods is that radial migration of non-adherent effector T lymphocytes can be measured in co-culture with adherent tumor cells. This has far-reaching implications for patients with primary or metastatic brain tumors because allogeneic cytotoxic T lymphocytes that are modified to express a prodrug activator gene such as cytosine deaminase are being tested as a multimodal approach for eliminating tumor in the brain.
Generally, individuals new to this method will struggle at establishing adherent tumor cell monolayers due to the heterogeneity of tumor cell growth and adherence characteristics.
Visual demonstration of this method is critical as the steps for generating an even tumor monolayer and an effective sedimentation of non-adherent allo-CTL require an experienced understanding of the characteristics of both cell types.